Hi, my name is Crystal, and the goal of my project is to see which genes are expressed when a sponge acquires its symbionts.
Hi, my name's Annie, and I'm looking at sea monkeys under an ultramicrotome.
Hey, I'm Brandon Sinowitz. I'm a biology student here at University of Richmond, a rising senior, and I've been working in a biology lab for the past 2 years doing research on sponges, particularly a sponge gene, and the protein that this gene codes for. What I like about research here at University of Richmond is not only the fact that I get to do essentially graduate level work in an undergraduate institution. I mean, we don't have postdocs or graduate students. We have essentially undergrads doing real lab work. You know, we're doing the experiments, we're designing the experiments, we're troubleshooting the experiments, which is pretty cool. It's a really good experience.
Argentina, Buenos Aires.
Hi, I'm Rachel,
Hi, I'm Sandhya.
and Sandhya and I are working on a joint project. We're studying polyglutamine diseases in fruit flies.
Hi, I'm Monica, and I'm studying about proteins that have to do with Alzheimer's.
Argentina.
So the thing that I think is really nice about this department is the fact that all of the tenure-line faculty in physics are research active. I mean, we're all doing experiments, taking data, writing papers, going to conferences, and we have to do all of this with students, so students really get to be a part of this. So we do a lot of different things. We have a couple people doing nuclear physics, we have someone doing astrophysics. My own work is in microscopy with an atomic force microscope, and a lot of my work dovetails with people doing things with electronics as well. So there are a lot of different opportunities for students who want to actually be a part of a research group and really act as if they're graduate students as undergraduates. So this piece of equipment here is an atomic force microscope. This is capable of resolving things about a nanometer in size. This instrument actually cost about $180,000, and I use this regularly with my students for looking at polymers. Thickness would then degenerate into islands and holes, and you get the plateau formation.
Hi, my name's James and I do genetics research on Shigella, and I'm using the scanning electron microscope to look at human colon cell samples that I've infected with Shigella.
The laser scanning confocal microscope, which has just been shut down. And with this microscope, you can optically section through samples and then do a 3D reconstruction of that material. It is the newest microscope that we have, and it's also the newest technology that we have. This is a transmission electron microscope. This is skeletal muscle from a tadpole, from Xenopus. This is a sample that was prepared for the biological imaging class. And to give you an idea of the resolution of the SEM, these little dashes in here are actually bacterial cells.
I'm Michael Leopold, an analytical chemist at the University of Richmond, and we're here in the NMR facility in Gottwald Center for the Sciences. NMR is nuclear magnetic resonance imaging. It's the same as people would see in MRI if they get their knee hurt or something like that. And at the University of Richmond, we're lucky to have 4 NMRs. We use it here to learn about the chemical structure of the compounds we've made or think we've made. And the unique thing about the University of Richmond is we encourage a hands-on research experience with our students as early as their first year. So as freshmen, they can be using state-of-the-art instrumentation that we have here. And it's all them doing it. They prepare the samples, they analyze the samples, they run the instruments without our help. And it makes them very independent scientists, and that's what we like.